Hey, in this video, I'm going to show how to use Matlab to process electromyography EMG data. And I'm just going to demonstrate. And in class, we can talk in more detail about this, but I want to give you a sense of how we process EMG data. Let me bring up a program we use, which is Matlab. And bring it up. So I'm not going to go into details on the coding, but I have offered coding seminars before, and students that want to know more about how to write custom programs to process data would take that type of course, and we would learn you would learn MATLAB, and then be able to process data very quickly. You can use Excel to do almost everything here. Working in Excel is so much more time consuming than doing something in MATLAB. The files I'm opening are data sets that were, I believe, 20 seconds long, recording at 2,000 samples a second. So 20 seconds times 2,000, that's how many rows that you would have in an Excel file. It's a big file. But processing that type of data in MATLAB is very efficient, and you'll see that in just a second. So I've got the program. I've written all these. These are all unique code programs I've written for different projects. And one that I'm processing data on right now is a body weight support project. And it's not important to know what the data consists of, but what the project involves is running with different levels of body weight support. And we're measuring electromyography of key muscles, lower extremity muscles, during running. So the first thing that we do when the subject comes in and gets set up with the EMG is we do a maximum voluntary isometric contraction, MVIC. And what that allows us to do is identify a value that we would use to normalize all our EMG data. The odd thing about EMG is if I put sensors on the surface of the skin above a muscle, that amount of electrical activity I'm going to measure, the actual absolute value doesn't matter. Okay, I know that sounds odd. What we do to process EMG data, first step is we normalize the data to something. For example, on ground reaction force data, we would typically normalize ground reaction force data to body weight. And then we would talk about a ground reaction force being 2 times body weight. Well, that's what we do with EMG. But instead of a body weight, we use maximum voluntary isometric contraction and the electrical activity that's recorded during that MVIC. And what you see here is, I hope you're seeing it, this bar is over that, is time on the x-axis. And for MVIC, we only record the person doing an MVIC for about 5 seconds. So the data set itself is just over seven seconds long. And that's time is on the x-axis, and EMG in microvolts is on the y-axis. And again, these values don't really matter other than we're going to use them to normalize data. So all our EMG data will be percent MVIC data, just like percent body, or in terms of multiples of body weight or ground reaction force. And what we do in our lab is we look at the data. Now, this file has already been full wave rectified, because remember, EMG will have both positive and negative values. And that just simply tells us the direction the electrical signal is moving relative to the sensors. Either the signal is moving towards the sensors or it's moving away from the sensor. But to process the data, one of the first steps we do is do a full wave rectification. We make everything positive. And so that's what's illustrated here. And then for identifying the electrical activity for MVIC, we identify the greatest 1 second average within that roughly 5-second MVIC. So someone contracts, we record, and then we go in and find the greatest 1-second average. How do we do that? Well, there's code that I've written that we calculate the average for the first second, and then we move the data over one, or move the window to average over one data point, and we calculate another 1-second average. And then we move the window over another data point and calculate another one. So you can think of what's happening here. You see this red bar right here. That's a 1-second bar. That bar is moving throughout the data set. And then we just go and find the greatest 1-second average amongst all the data that were calculated. And that's what's illustrated here. So you can see it doesn't capture this single peak. Other labs actually identify the greatest single peak. I don't do that because this peak happened in 1/2000th of a second. And I want to have a signal that represents muscle activity more so overall, as opposed to a single data point. Okay, so that is the rectus femoris MVIC. Then we go on and we continue the program. And there it's running. Okay, and then the tibialis anterior. Again, finding the greatest 1-second. In this case, it was right in the beginning of the MVIC. And I like to plot this just to get a sense of where that MVIC was the greatest. That just helps me visualize the data, make sure that there aren't any errors in the MVIC file or something odd happening. And we did 4 muscles. So this is the bicep femoris muscle. Here's the greatest 1-second average all the way over here. And for this type of MVIC, when I'm inspecting the data, I'm looking for a quiet period of muscle activity, and then we're in the MVIC part, so I'm looking for lots of activity, and then we tell the person to relax, and then we like to see this quiet period after the MVIC. And here's the greatest 1-second average for the gastrocnemius. Once that's done, I plot out the data. Now, this is already partly processed because this is all full wave rectified. I didn't plot out the raw data in this case. Raw data would be showing both positive and negative signals. Full wave rectified is everything is positive. And one of the things we do when we look at the data is we do a visual inspection of the data. So in this top plot, this is rectus femoris. The next plot is tibialis anterior. The next plot is biceps femoris. The next plot is gastrocnemius. And then an acceleration signal down here. And then X is time, and it's the same time axis for each of these. So remember, each sensor has accelerometers associated with it. And what this acceleration signal on the bottom represents is a compilation of all the acceleration signals in a resultant direction. And I've done that because I use that to identify the time that the foot is striking the ground. And then from there, we're able to identify strides during running. So when I see this plot, one of the things I'm looking for are some nice bursts of EMG and some quiet periods of EMG. So this is rectus femoris. And clearly, rectus femoris is going to be active right after foot contact. And that's going to correspond to an acceleration peak down here. The rectus femoris can also be active right before contact, too, because you're preparing the muscle for ground contact. And so you'll see later on that that peak of activity doesn't necessarily occur at contact. And then the other thing I look for is potential characteristics of noise. So you see this spike right here. That's probably a movement artifact of the sensor, because it's uncharacteristic. It just sort of comes out of nowhere. Here you see a burst, 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 and then this one happens somewhere between the regular bursts. So some type of noise entered the signal. So we got to deal with that when we're processing the data. Sometimes the way we deal with it is we avoid that noise and we start analyzing the data after a noise artifact like that. And sometimes it's hard to distinguish between what's noise and what's real signal. And so we have these different smoothing routines that we do. And in this case, I'm going to go ahead and smooth the data using a low pass filter. And I believe I have it set for a 4 hertz cutoff because I'm really interested in this project, the pattern of muscle activity. I'm not actually really interested in the magnitude so much, but I'm interested in the pattern. And so now you can see the signal is so much smoother. Okay, we've not seen that real quick oscillation of the signal. That's by design because we're interested in how the muscle is behaving from a coordination perspective. All right, so now I inspect that. I make sure that that is reasonable, and then I continue on with the program. And at this point, what I'm going to do is identify the beginning and ending points of a stride. And you see this very distinct peak. So now, oh, that says Elgon, because that's from the older iteration of the project. This is acceleration data on the y-axis. What I'm going to do is identify that peak right there that corresponds to a foot strike. We see a large acceleration spike right as the foot contacts the ground. And the way I've written this program is we identify 4 of these peaks. And because I'm lazy, then using those 4 peaks, I can find the remaining 15 peaks. All right, and so we actually, excuse me, 16 peaks because we're actually identifying 15 strides. And to get 15 strides, we need to start a stride, which would be 0, and then we go to 16 consecutive peaks. All right, and then after finding those peaks, we go in and pull the data out for each stride, and I compile them here. And so here's rectus femoris. This now on the x-axis is percent stride. Okay, and meaning that we normalize the time to be 100% between foot contact to the next foot contact to the next foot contact. And then extracting the data for the rectus femoris for each of those strides and plotting them here. So there's 15 lines here. And what you can see is that of those 15 strides, they're almost all similar. Maybe there's some differences right around here, maybe some subtle differences in here, and so forth. And we look at that for each of these muscles. This one, the TA, there's something odd is going on here with a couple of strides. So that might be something that would draw my attention to say, okay, what's going on with this one? Did I select the wrong data or what have you? But nevertheless, I've got more variability in how the TA is being recruited during running for here. Here's the biceps femoris, very similar pattern for all strides, and same thing for the gastrocnemius, maybe something a little different happening near toe off. And then this is just the acceleration signal down here, just making sure that I'm getting similar patterns for each stride. Then I create what's called an ensemble plot. I take those 15 strides and I average them and I create a standard deviation. And so now this blue line represents the mean pattern for the rectus femoris during a stride. Again, this x-axis is percent stride. And then same thing for the TA, the biceps femoris, and the GA. And you'll notice that some of that, those couple of strides, they sort of get washed out when you start averaging multiple strides. That's by design because we are trying to make sure that we're representing overall the muscle activity during running. And maybe one stride isn't different enough to really draw our attention to. But nevertheless, that's what we have now for our plot for muscle activity patterns. And then what the program does, it saves all that information, actually averages the data across the 15 strides, calculates root square mean, does all of that, and then plops those results into a file, which I'm not going to show you here. And then I take that file and I open that up in Excel for running my statistics. I actually run my statistics through SPSS, but via Excel. And then I move on to the next condition. So that was subject one, or subject, I think it was subject 9, condition 1. This project actually had 6 conditions, but I'm only going to show 2 for the purposes of this video. But now I've moved right on to C2. Notice that we're not going to do the MVIC now because that was already done. And given it was done, there's no change in lead placement. Everything's the same. I can use that same normalization value to normalize these data too as well. So then I normalize the data and smooth it. And I get this data set. And I'm looking for patterns that are repeatable. I'm inspecting the data qualitatively. And then I go in and I identify those 4 peaks. So I can pick any of them along here. They just need to be consecutive peaks. Yeah, it's a funny acceleration peak, but it happens at the same time. I'm actually not interested in the magnitude of the peak. I'm just using that for a demarcation for the beginning and ending point of a stride. And then once again, I get these 15 strides of muscle activity. That's a nice, very consistent pattern across the 15 strides. TA, fairly consistent, something odd going on here with a couple of strides. Biceps femoris, relatively consistent. Gastrocnemius, again, a couple of odd ones near the toe-off portion of a stride. Or actually, yeah, sorry, this would be right before contact, because a stride will include stance phase and also flight phase. And then we get the ensemble plot. And this is what we would use ultimately as we continue our analysis of muscle activity patterns. And again, during that, the 15 strides, pull out the average EMG, the root mean square EMG, any other dependent variables that I want to generate from the time domain analysis, and I plop those into a summary file. And then the program ends, and that's it. So if I did that in Excel, doing all those steps, that would probably take me at least an hour per subject. To do it during Matlab and talking, it is whatever the length of this video was. Very efficient way, so that what's nice about that is, as the researcher, you can spend more time inspecting the data and trying to understand what's going on with the data as you're processing the data. Okay, that's Matlab. All right, thanks.